Hi everyone, my name is Sloan. I'm 18 and I'm an osteo warrior. I'm also an amputee. I'm here to announce this lovely moderator. Dr. Laura Davis is the sarcoma program director at the Knight Cancer Institute in Portland, Oregon, where she treats patients with sarcoma and conducts osteosarcoma clinical and translational research. She received her undergraduate degree at Wellesley College and her MD from Oregon Health and Science University, OHSU. She returned to the East Coast for residency training before completing her oncology training at OHSU. Dr. Davis is board certified in both medical oncology and pediatric hematology oncology.
All right. Last panel of this amazing conference. Thank you all for being here. And thank you, of course, to Anne and Christina for putting together this panel and inviting me to moderate. I have the honor of sharing the stage with some powerhouse researchers in the field of molecular targets and targeted therapy in osteosarcoma. So I'm going to start out with brief introductions, and they're going to take it away and blow you out of the water with what their research is doing. So Dr. Eunice Lopez Fuentes is from UCSF, where she is working in the Sweet Cordero lab. There seems to be a representative on every panel during this conference, and I love that, representing the excellent work that's happening there. So she's a postdoc fellow. She completed her master's in biochemistry and molecular biology in Mexico, and she also got her PhD in molecular biology there. And she is working on defining the mechanisms of epigenetic gene regulation in osteosarcoma, and is going to tell us a bit more about that. Dr. Tony Musters is a PhD in vet from the Ontario Veterinary College at the University of Guelph. And he's an associate professor working in the departments of clinical studies and biomedical science. And he has a clinical oncology practice within the veterinary school, and his research program uses comparative oncology approaches to translate discoveries from preclinical to clinical applications in humans. And he's investigating, or he has a focus on osteosarcoma as well as angiosarcoma, melanoma, and bladder cancer. Next, we have Miriam Azim, who is a PhD candidate at the City University of New York. She completed her undergraduate work in India and is now, she is now pursuing her PhD at the City University of New York, and she is going to talk to us about how she may be able to repurpose a drug that is currently used for ALS to potentially treat osteosarcoma. And we are going to give Miriam an extra special welcome and thank you because this is her first major conference presentation, so we are going to give her the love. Tanya Heem is next up. She is at the University of Pittsburgh in the labs of Drs. Kurt Weiss and Ines Los. She has her bachelor's in animal science from Penn State and a master's in biology from Youngstown. She actually worked as a vet tech before coming to the University of Pittsburgh, and she is investigating the androgen expression and the way to maybe repurpose some prostate cancer drugs for the treatment of osteosarcoma. Those first four speakers are kind of looking at some emerging molecular targets that are a little bit earlier on in the preclinical realm. And then our next three speakers following that, we'll have a break kind of in between the first four and the last three. The subsequent three have a little bit more involvement on the clinical realm thus far. So Dr. Kristen Van Heist is from Rainbow Babies at Case Western. She is a physician researcher and an assistant professor in pediatric oncology at Case Western. And she is working she works with Dr. Alex Wong, who was just up here, and she is also in the lab, and then she's also PI of some clinical trials that she will be talking to us about. And then Dr. Jason Eustein is an MD PhD from Emory University and is a physician scientist and pediatric oncologist there, and he is testing 2 drugs that are currently in trials for osteosarcoma, but he's looking at combining those and really understanding their mechanisms and how they might be effective for osteosarcoma. And then Dr. Nadia Omolchenko is a research fellow and a radiation oncologist by training at the Sarcoma Oncology Research Center in Santa Monica. And she is going to present some work that Dr. Erlinda Gordon is well known for. Dr. Gordon is a director at the Sarcoma Oncology Research Center and medical oncologist. And their presentation today is on Delta Rex G and gene therapy for some exciting new targets in osteosarcoma. So, without further ado, oh my gosh, I already went over? Oh, this is terrible. So I just ate up 30 seconds of your time, but I will try to keep everybody on time and use some intimidation tactics from over here if you're going over, which you guys should have done.
Thank you, Dr. Davis, for the introduction. I also want to thank Christina and the team for organizing this amazing conference. My name is Eunice Lopez Fuentes, as already Dr. Davis said. I'm a postdoc in Alejandro's Sweet Cordero lab. And today I'm going to talk about the epigenetic landscape in osteosarcoma. So I want to start introducing epigenetics. So a classic example to explain epigenetics is identical twins. So in identical twins are from a single egg that is fertilized by a single sperm and then it splits in 2. So each twin has exactly the same DNA information, the same genes. However, they are still have difference. For example, here in twin A, this gene is expressed because of the conformation of the regulatory region. It is open and accessible. And in twin B, this gene is not expressed because the conformation is closed. You can see the nucleosome arrangement in the figure. So in cancer cells, you have mutations in the DNA, and then the DNA is condensed and wrapped around the nucleosomes and organized in the nucleus. So you can have 2 different conformations. A condensed conformation where the genes are not expressed because the machinery of the cell cannot have access to this DNA, and an open conformation where the genes are expressed because the machinery of the cells can have access to the DNA. So I'm going to talk about these 2 different states: on, which is open and accessible, and on, which is closed and condensed. So epigenetics plays an important role in cancer since it can turn off a tumor suppressor, but also turn on a tumor helper. So in this project, we are looking for patterns of all the open genomic regions to define different cellular states. We have 4 goals. Defining the epigenetic cell states in osteosarcoma. We want to define the master regulators that are orchestrating the gene expression programs. We want to identify and validate the differential drug response in vivo. And lastly, define the markers to predict the epigenetic cell state in patient samples. So we perform a TAC-seq, which is the technique that we are using to generate this data of chromatin accessibility. We have 45 osteosarcoma samples. We are doing 2 replicates per sample. And a TAC-seq basically allows to look at these open genomic regions because we are using an enzyme that is coding only the accessible genomic regions. So this cohort contains cell lines, the ones that are commercially available, PDX cell lines that were generated in our lab, PDXs, and patient samples. We analyze all these chromatin accessibility data, and here is the PCA in an unsupervised way. And you can see how Most of the commercially available cell lines are clustered here to the right. They are in pink in this PCA. You can see then the PDX cell lines in blue, which the distribution is wider, suggesting that there is a higher heterogeneity regarding chromatin accessibility. We also have the xenograph in orange and the patient samples. Using different approaches, we define two different clusters. Cluster 1 here, or the cellular state 1 here in green, and cluster 2, or cellular state 2 here in orange. So the first question that we asked after defining these two different cellular states by chromatin accessibility was, can we see differential drug response between these two different cellular states? So we took advantage of this data set that was generated. It's a drug response data set that was generated across 40 different targeted agents. We do the assignment of the epigenetic cluster that we defined previously using a PDX line panel, and then do the analysis. We found that one of the cellular states responds to a MEK inhibitor, and the other cellular state responds to aurora kinase B and aurora kinase A inhibitor. So this drug response data set was generated in vitro. So we wanted to validate this finding in vivo. We are using a subcutaneous model. So we took a PDX line from cluster 1, cellular state 1, and we saw that it's responding to aurora kinase B inhibitor, but it's not responding to the MEK inhibitor. Then we do the opposite experiment. We took a PDX line from cluster 2, of cluster 2, or cellular state 2, and we found that it's responding to the MEK inhibitor, but it's not to the aurora kinase B inhibitor, which is validating what we have seen in vitro. Then we define these two cell states using bulk data. So we know that osteosarcoma shows a lot of heterogeneity. So we ask if we can find more than one cellular state in one osteosarcoma tumor. In order to address this question, we treated a xenograph with aurora kinase B inhibitor, MEK inhibitor, and then the combination. And we saw that the response to the combination is even better than the response to the single drug. This is suggesting that we can have these two different cellular states in one tumor. So we analyzed single-cell RNA-seq data of 4 PDX cell lines. So now we have this single-cell resolution to answer this question. And then we do an inferred assignment of the cells using a gene signature that was previously generated using chromatin accessibility data. And you can see here, like, the cells, the green cells are cluster 1-like, and the orange cells are cluster 2-like. But we also have these gray cells that we think this is the intermediate state between these two cellular states that we are defining by bulk ATAC-seq. So you can see here the enrichment of this, the quantification of this analysis, and we found that the cluster 1, we do see an enrichment of cluster 1-like signature, but for cluster 2, we saw like a mix of these signatures. So another advantage to use this approach, to generate this chromatin accessibility data, is that you can find the transcription factors that are bound to the DNA, because the transcription factor is protecting the DNA from the enzyme that is coding the open genomic regions. So we did the analysis and find that for cluster 1, we found rungs 3, rungs 2, all these transcription factors that most of them are involved in development. And for the other cellular states, cluster 2, we found most of the AP1 members of this complex, the AP1 complex. So then we ask if these master regulators are co-expressing or are acting together. So we analyzed gene expression data, and we did a correlation metrics, and saw that the transcription factors from cluster 1 are expressing together, are co-expressing. And same for the transcription factors of cluster 2. And we do see this anti-correlation between these two sets of master regulators. Then we took advantage of a different data set, a completely orthogonal data set, which is the acetylated regions of these PDX cell lines. And in an unsupervised manner, we saw that when we color code these two clusters that we defined previously, the 27 acetylation histone mark is able to separate these two cellular states, which is suggesting that these epigenetic clusters are driven by enhancer regulation. So it is really exciting that this completely orthogonal data set is validating our chromatin accessibility data. Then we asked what transcription factor motifs are enriched in these acetylated regions. And we found that most of the transcription factors that we saw in these two clusters, they are mostly the same that the ones that we are defining by chromatin accessibility. So in summary, we are defining two epigenetic clusters in osteosarcoma with a distinct transcription factor set that is orchestrating gene expression. And we saw that these two cellular states are showing differential drug response. I'd just like to thank our lab, Sweet Cordero Lab, and particularly Liane, Andrew, Alex, Elizabeth, and Maria, our collaborators, and of course, the funding. I would be happy to take any questions in the discussion panel. Thank you.
Okay, thank you to MIB agents and Christina for the invitation to be able to share with you some of our preliminary data today. And a special thanks to the Travel Fund. Because of Isaac, that was able to bring me here. And even though Isaac Chung's family wasn't able to join us, I've been proudly wearing this pin, and appropriately it contains a picture of Isaac and his dog. So how appropriate to be able to bring me down from the Ontario Veterinary College to speak to you today, and I'm honored to do so. I have no disclosures to make, and of course, as a veterinary oncologist, I work with this canine naturally occurring translational model of osteosarcoma, which we've heard so much about over the last 2 days that I can move on to the next slide. We're all very familiar with its properties and how it might be utilized as a model for human osteo research. So our project, and this is really the project of Andrew Poon, who's a PhD DVM student in my lab, and I wish he was able to come here to present it himself. He's an intern now, so he's rotating on nights and ER duty and things like that. He was really interested in taking our lab into a metabolism focus. And this is based around, this background with the Warburg effect. And so the classic definition of metabolism in the context of normal differentiated tissues, if you go back to high school biochemistry and you think about in the presence of oxygen, the generation of large amounts of ATP in the mitochondria. Conversely, if you think of exercise physiology and lactic acidosis, when oxygen is depleted, you have this production of lactate, and how the Warburg effect defines this preference for the utilization of lactate to generate a relatively small amount of ATP independent of the amount of oxygen that's available, this addiction that tumors have. And in the last 5 or 6 years, it started to become known that this seemingly byproduct lactate of this Warburg effect has a role to play in metabolism. And this Nature paper from 2017 demonstrates how glucose can feed into or lactate can feed into the TCA cycle. And so you see the classic example of glucose in the cell, lactate which can then go into the bloodstream and back into the cell for use in the TCA cycle. And these investigators utilized labeling of lactate and other things to demonstrate that intermediates like malate and succinate in the TCA cycle came from this lactate. And so it's sort of redefining this paradigm or our use for the Warburg effect with all this generated lactate. And so Andrew really wanted to investigate this kind of thing further as it might apply to osteosarcoma. So the objective was to essentially profile by comparing the glycolytic and oxidative and alternative fuel metabolism in both canine and murine osteosarcoma cells, to take a comparative approach, and ultimately in human cells as well. and compare those with mesenchymal stromal populations, the osteoblast populations, and then narrow the focus a little more to superimpose other things that may be affecting them, like the extracellular environment stiffness, hydrogels, for example, or the influence of hypoxia into the way these metabolism was going. So we started with preliminary data in 12 cell lines, 6 canine, 6 murine. So these color-coded examples kind of carry through the rest of the talk. The canine cells were mesenchymal stromal cell populations, and then the light blue are primary osteosarcoma cell lines, and the dark blue are metastatic osteosarcoma. And our mouse model was a transgenic Crelox Ostrix-1 driven P53RB knockout that allowed us to have paired primary and metastatic pairs from those murine models, as well as a very familiar K7M2 metastatic murine cell line to study. We leaned heavily on the Agilent Seahorse platform as a way in real time to get an assessment of glycolysis and oxidative phosphorylation through a semi-high throughput way of looking at oxygen consumption and acidification in these cell lines, and allowing us to do a standardized stress test for both glycolysis and oxidative phosphorylation to quantify things like glycolysis, glycolytic capacity, and reserve ATP production and things like that. So to get into the data, this is the glycolytic platform across the cell lines, so the blue being the canine cell lines and the red being the murine for glycolysis capacity reserve. The take-home message from this slide is that essentially the metastatic osteosarc cells were highly glycolytic in comparison to both their primary and MSC counterparts, with one notable exception being the Abrams cell line, which again, those working with canine osteosarcoma may be familiar with. It's categorized as a primary, but a very aggressive cell line in its own right. But across the metastatic cell lines, we were seeing this large glycolytic capacity. And then doing the mitochondrial stress test to look at oxidative phosphorylation, a similar result was obtained where the metastatic osteosarc cells had this extremely oxidative profile and ability to be very flexible. Again, in contrast to those primary cell lines. And this represented a bit of a metabolic gap in explaining both the glycolysis and the respiration, and was that lactate utilized from the glycolytic production. So the final part of this profile was essentially looking at profiling these in a 2x2, where if they're highly glycolytic, they'll be down here, highly acidified or highly aerobic. The metastatic cell lines, with that Abrams exception, were in the energetic quadrant, and then were highly flexible when stressed with oligomycin and FCCP. So the utilization of different constituents like glucose, pyruvate, and glutamate was the next step in Andy's work, and he did this both in normoxia and in 1% oxygen hypoxia. The take-home message from these were that most of these were utilized to increase viability in the hypoxic setting, both in the canine, which is from this slide, and then in the murine, with the one notable exception being glutamine in the hypoxic environment had a lesser viability over time. But his next work was in the matrix stiffness arena. And so these were calibrated hydrogels to 2 kilopascals to try and represent the lung microenvironment, and 100 kilopascals for the bone microenvironment, compared to normal cell culture plastic. And we grew these for 7 days in these environments. Took the cell pellets to analytical lab at sick kids, where they were able to perform the mass spectrometry for the glucose and metabolite intermediates and central carbon metabolism. And then grouping those by Z-score, if you compare the culture plastic to those in the soft and then the stiff hydrogels, 9 of the cell lines in that soft environment had increasing levels of those metabolites in general sense, and 4 cell lines in the stiff group. Again, representing whether that was upregulated in the metastatic environment. Now, to look at this as a proportional abundance representation, again, color-coded so that the glycolytic-oriented components are in shades of red and the oxidative in shades of blue. It was really lactate and malate that were the real predominant metabolites across the board in these cell lines, both canine and murine. And then we wanted to look at what this might mean. Is it speak to the utilization of lactate? And to try and facilitate timing, I took a few slides out of here that really looked at this lactate utilization. So this is the bare utilization and the oxygen consumption when these cells, both mesenchymal stromal and cancer, were fed lactate. We were able to abrogate this with interfering with that malate aspartate shuttle, as well as interfering with pyruvate uptake, which is the presumed way lactate would feed back into the mitochondria to produce oxidative phosphorylation. So indirect evidence here using the seahorse that we were getting lactate utilization in these cells, both in cancer and in the stromal cell. Now, another way lactate travels back and forth into the cells is through the monocarboxylate transport system, MCT1 and 4, targetable system. When we looked at viability in those cells on the left, as a single agent, we weren't seeing a lot of changes in viability. In addition to chemotherapy, carboplatin, we did see changes in the cancer cells compared to the stromal cells and reductions in oxidative oxygen consumption when targeted with the MCT1 inhibitor from AstraZeneca or serosingapine, which targets both MCT1 and MCT4. In the murine cells, it was a little cleaner. Again, preliminary data with just these 12 cell lines showing that the lesions in the lungs, the 494L, with both serosingapine and AstraZeneca drug, were reducing oxygen consumption and reducing viability in addition to carboplatin. So Andrew drew this really nice schematic that also, because I could never do a biorender that looks this nice, shows the utilization of lactate, and even in here, the way it would shuttle through the malate aspartate pathway and presumably go through pyruvate. I think that speaking to the first Nature paper, it looks as though we've got the relevance of lactate utilization in osteosarcoma. And the second takeaway message is this new recent Nature paper that's come out showing that when you compare primary and metastatic, primary tumors in these GEM models of pancreatic and lung also have very low TCA flux and ATP production in the primary compared to the met. And so that other take-home story is, again, the importance of really studying what it is in the metastatic setting as the relevance for targeted therapeutics. So thank you to every granting agency. This is a picture of Andrew in the middle who did the bulk of this work, and I'll be happy to take questions in the break.
Good afternoon, everyone. Thank you, Dr. Davis, for that lovely introduction. Whew, already feels like I'm defending my thesis right now. All right, let's get started. All right, thank you, MIB, for giving me this opportunity. This is my first Factor conference, and I'm delighted to be here. Definitely inspirational. The journey has been past 2 days. It's amazing. So I'm glad to be here to represent my data. I'm a PhD candidate in Mahajan Lab, and I am investigating the role of EGR1 in riluzole-induced apoptosis and osteosarcoma. So the goal of our lab is basically repurposing riluzole for osteosarcoma treatment. So riluzole, which is currently used for ALS, it's available in the market under the name Rolotech, and we have this well-characterized profile for riluzole coming to toxicity. And the way it works in ALS, it blocks glutamate release and thereby affects glutamate receptor activation. So it's basically blocking the autocrine signaling of glutamate. And what do we know about glutamate? So glutamate is needed in bone cells for differentiation and development. And from previous studies in melanoma and gliomas, riluzole has actually shown to slow cancer progression. So this is a very comprehensive image that we have put together to show how riluzole causes cell death in various cancers, but I'm not going to go through everything. We are very focused on how riluzole actually affects this blockade of glutamate receptor activation. So going further, let's take a closer look. Before taking a closer look, I just want you to focus on YAP and P73. So YAP is yes-associated protein. It binds to many transcription factors and modulates its activity. And coming to P73, it's a homolog of P53, and it has tumor suppressor activity. But when they come together, binds onto the promoter region of their target genes, it can actually induce cell death and osteosarcoma. So our lab recently elucidated a mechanism of riluzole-induced apoptosis in osteosarcoma. So what we have seen is when we treated these osteosarcoma cells with riluzole, they undergo apoptosis or cell death. And how does it happen when these are treated with riluzole? There's increase in ROS production, which is reactive oxygen species. And once there's increase in ROS, of course, there is DNA damage by which c-Abl kinase gets activated. And this activated c-Abl kinase phosphorylates YAP inside the nucleus. And this phosphorylated YAP, along with P73, binds onto the promoter region of BAX, which is a pro-apoptotic gene, and increases its expression. So once BAX expression increases, it induces apoptosis or cell death in osteosarcoma. So when I was working on this with Dr. Mahajan, I was very interested in looking at what other genes or what other proteins that are involved in this complex that you see here. In this complex where we have YAP, P73 binding onto the BAX promoter region, and I was looking at different YAP interactors, and EGR1 caught my attention. And when I was reading about EGR1, early growth response 1 gene, there were not a lot of studies in osteosarcoma. But when I looked at other cancers, there were a lot of studies which, where they conducted studies, and it showed that it actually acts as a tumor suppressor in other cancer types, but not in osteosarcoma. Oops. So coming to EGR1, what is EGR1? It's early growth response 1. It's basically a type of zinc finger protein. It's a transcription factor, and it recognizes the GC-rich sequence. You can see here it binds using the GC-rich sequence onto the promoter regions of its target genes. So in normal cells, it's basically regulates the ECM, is involved in cell proliferation. But when it comes to abnormal cells, when it is overexpressed, it actually causes cell death. So let's look at the different stimulants. So there are different factors involved that actually increase EGR1 expression inside the cell, one of them being ROS, and as we have seen previously, that riluzole increases ROS. Now the question was, in osteosarcoma, does ROS actually increase this EGR1 transcription? And then when we looked at its target genes, you can see that EGR1 binds to target genes. One of them is BAX. And we have seen that riluzole increases BAX expression. That kind of interested me. And so I came up with the hypothesis that EGR1 plays a role in riluzole-induced cell death. Now the question was how it is involved. So I looked into some of the papers. So there are very few papers on EGR1 in osteosarcoma. And one of the very few papers that I came across where they tested MAP therapy drugs in osteosarcoma to see the EGR1 expression. And what they did, they looked at the basal expression level of EGR1 without even treating them. So what you see on the left is the EGR1 expression level, basal expression level compared to the normal human osteoclast. And they saw that it's suppressed. When they treated with the MAP therapy drugs, they saw that there is increase in EGR1 expression. Now, this kind of was really interesting. So I went ahead and I replicated data. I just wanted to see the basal expression level in all these different cell lines compared to the normal human osteoclast. And we see that the expression is suppressed. Of course, now the question was, how about we treat osteosarcoma cells with riluzole, since it's already established with MAP therapy drugs? So this is a very preliminary data that I've been working on. So you can see that with all of these cell lines, EGR1 expression went up when we treated with riluzole. after 1 hour. And this is at mRNA level that we have seen that riluzole increases EGR1 expression. Then, of course, we went ahead to see the protein expression level. And with riluzole, we saw that EGR1 protein expressed pretty highly. And now the next question was, does riluzole increase ROS and osteosarcoma? All the cell lines, because previously, when we were working on the paper, we only worked with one cell line, the LM7 cell line. So now I wanted to first see if ROS is increased in all of these cell lines. So we do see that there is increase in ROS across all the cell lines. Now, the question is, does this riluzole-induced ROS increases EGR1 expression? And again, this is a very preliminary data that I've collected, and where we see that there is, so what we did, we treated the cells with riluzole, we saw increased expression of EGR1, and then we treated the cells with NAC, which is N-acetylcysteine, which is an antioxidant that opposes ROS production. And then you can see both at mRNA and protein level that there is basically decrease in EGR1 expression when you add NAC, representing that ROS is a direct stimulant of EGR1 expression. So I still have to perform this in other cell lines. So moving forward, taking all the in vitro data that I have, wanted to move into in vivo studies, and we have 2 models for that, and one of which is the metastatic mouse model. The other one is the PDX cell lines, patient-derived xenograft lines. Coming to the mouse models, so these mouse will be injected with the osteosarcoma lines, and then they will be treated. There will be control group and treatment group. And then once we resect those tumors, we'll be examining the tissues for EGR1 expression. And with the patient-derived xenografts, we have collaboration with Dr. John Healy at MSKCC. So we'll be receiving the cell lines from his group. And we'll be taking these cell lines, growing them in vitro, and then examining the EGR1 expression in these cell lines. Again, very preliminary data. This is a PDX cell line, which I just performed before coming to the conference. And so what you see here is both at mRNA and protein level, you see EGR1 expression when treated with riluzole. So what we expect next is we expect to see an increase in EGR1 expression upon riluzole treatment in both of these models. In the long run, the bigger picture here is to understand how EGR1 acts in osteosarcoma, because there are not a lot of studies. EGR1 there are a lot of studies in breast cancer where it acts as a prognostic marker, and with osteosarcoma, we don't have that information. And then if everything comes out to be well, and if riluzole is effective in the treatment of osteosarcoma, and if EGR1 over all the cell lines, we see the same data being reproducible, it can act as a therapeutic biomarker. So with that, thank you so much. I would like to thank my PI who gave me this opportunity to be in her lab, perform this research, and my lab members, definitely our collaborators as MSKCC, who generously was ready to collaborate, give the PDX cell lines to perform this research. And thank you everyone for listening. This is the last panel. So thank you so much for giving this opportunity to me. Thank you, Christina, for actually organizing this and accepting me here. Thank you so much, everyone.
A quick reminder to everybody to use Slido. It's ready and available for you.
Okay, before getting started, I just want to echo what everyone's saying is thank you so much, MIB agents, for hosting this conference. It's been absolutely fantastic. It's my first factor, and I've just met so many amazing people, and just it's been so inspiring, so thank you. I also want to recognize and thank the Myrovalitis Trust for giving me a travel award to be here with you today. Okay. Our lab's main focus is studying how we can find better treatment options for patients with metastatic osteosarcoma. I'm not going to go over the reasons why metastatic osteosarcoma is a problem. We've heard that a million times, and we knew that most likely before coming to this conference. But because most of metastatic osteosarcoma occurs in the lungs, we decided to sequence matched patient primary and recurrent tumors compared to their respective lung metastases. And that's what you see here in this heat map. On the left side of the heat map, under the green bar, are 4 different patient primary and recurrent tumors. And then under the red bar on the right side is their respective lung metastases signatures. So this led us to believe that metastatic osteosarcoma is a different disease than primary osteosarcoma, and the treatment that we use to treat lung metastases should be specific to the lung. One target that our lab has studied extensively, Kurt Weiss, my PI, he's actually studied it since he's a fellow, was aldehyde dehydrogenase 1A1, or ALDH 1A1. Essentially, this enzyme helps cells withstand stress. It is part of a larger family of enzymes as well. High ALDH activity has been conferred with increased chemoresistance in osteosarcoma patients and also increased metastatic potential of osteosarcoma cells. Fortunately, there are several ongoing clinical trials that are using ALDH inhibitors right now. Androgen receptor, or AR, is a sex hormone receptor, and while Dr. Davis mentioned, it is more extensively studied in prostate cancer. Its expression has been noted in soft tissue sarcomas and also osteosarcoma. We decided, and also growth and sex hormones have been thought to play a role in the development of osteosarcoma, and this may help explain why we see a higher prevalence of osteosarcoma in males versus females. The heat map that you saw just a couple slides ago was made using RNA sequencing data, and this data was also very, very beneficial in helping us identify pathways that were upregulated in our primary, recurrent, and also lung metastases tumor samples. And very interesting to us, one of the pathways that was predicted actually predicted AR to be upstream from ALDH 1A1. So this led us to further investigate the expression of AR and how it relates to the expression of ALDH in osteosarcoma. We quantified the expression of AR and ALDH in a primary osteosarcoma cell line, SALS2, and a metastatic lung metastases cell line, LM2. And you can see in the figure on the left that unexpectedly, or not unexpectedly, as expected, sorry, the lung metastases cell line with high metastatic potential did have higher expression of the ALDH 1A1. But we also did see that the primary osteosarcoma cell line showed higher expression of AR. We saw this expression of AR both at the mRNA level and at the protein level, which you can see in the Western blot. So this led us to believe that metastatic progression may involve the regulation of these 2 targets. So we decided one of the best ways to further study ALDH and AR in osteosarcoma would be to create some stable knockdown cell lines. We first attempted to knock down ALDH and AR in SALS2 and LM2 using lentiviral vectors. And the figures you see here are the results from those transduction studies. So on the left, you see ALDH 1A1 knockdown in all of the transduced cell lines. And I just want to direct your attention to the, to the bottom black bar. Actually, both bottom black bars, that's our non-targeting control. And so on the left figure, you can see that it had ALDH % knockdown that was similar to the other transduced cell lines. I keep doing that. The other transduced cell lines that actually targeted ALDH. So it was having a nonspecific effect that we were seeing, and then we also saw an increased percent knockdown of AR in the non-targeting control. And kind of the cherry on top of all of this was in our lung metastases cell line. When we looked at the percent knockdown of AR, it seemed to not matter what the lentiviral vector was targeting. It just seemed to have a very similar increased knockdown of AR. So we felt that these findings showed that our targets may be responding to the viral infection in a nonspecific way. And so we decided to just follow the biology. And if we can't knock down these targets at a transcriptional level, then we can try doing it at a protein level. And as Miriam discussed drug repurposing, we really feel we're a translational lab. And so we really like to look at drug repurposing as a tool because it gives you over 19,000 different drugs that you can pull from and hopefully get to the clinic faster. And luckily, there are a few AR inhibitors. Enzalutamide is an FDA-approved AR inhibitor that's currently being used to treat metastatic prostate cancer patients or prostate cancer patients that are chemo-resistant. We used enzalutamide to treat our SALS2 and LM2 cell lines, and then we treated them for 24 hours and then assessed viability. Both cell lines did show sensitivity to enzalutamide. However, interestingly, LM2, the lung metastases cell line, showed to be much more sensitive to the treatment, while SALS2 had a much more variable response. Next, we tested enzalutamide's effect on osteosarcoma cell migration. And to do this, we used a Boyden chamber method. And if you're not familiar with the Boyden chamber method, I'm just going to explain it very quickly for those in the crowd. So what we did first was we pre-treated cells for 24 hours with enzalutamide. And then those cells were seeded onto a porous membrane. That's represented by the lime green bar right there. Sorry, I'm shaky. So the cells were seeded onto the top of the porous membrane, and then a chemoattractant media was placed underneath the membrane. Cells were given 18 hours to migrate, at which time the assay was stopped. Any cells that did not migrate were removed with a Q-tip on the top of the membrane, and the cells underneath the membrane were stained with crystal violet. The figure at the bottom left shows you the comparison between treated and untreated cells. And you can see that visually, enzalutamide decreased migration in both of our cell lines. And then we quantified the amount of crystal violet from these migrated cells and saw that also enzalutamide significantly decreased migration of both the primary and lung metastases cell lines. I do want to mention that I'm in repeat experiments of this to validate these test results, so stay tuned. Okay, so in summary, our lab's motto, as always, there's a tremendous need, and there's a tremendous need for patients with osteosarcoma lung metastasis, and we need to do better to treat those patients. ALDH1A1 and AR are inversely related in primary and metastatic osteosarcoma samples. And while we did have trouble knocking down these targets with lentiviral methods, they are both druggable targets. And regardless of the metastatic potential of the cell lines in this study, enzalutamide did seem to be a promising treatment. So with that, I just want to thank all the members of my lab at the Musculoskeletal Oncology Lab at the University of Pittsburgh, especially Dr. Kurt Weiss and Ines Losa, my co-Is of the lab. And obviously, I want to thank MIB again for the tremendous support that they've given us for this work and all the other funding sources that are noted there. Thank you for your attention.
All right, excellent. Thank you, speakers. We have a few minutes for questions. I can't see anything.
First question is going to be over here to your right,
Way over here.
far right.
Excellent.
I just had a quick question for Dr. Lopez Fuentes. Thank you for presenting all this data. I just wonder, looking at the transcription factors and the fact that state 2, I'm way over here, looking at the transcription factors and that the cell lines all tended to be state 2, could it be possible that state 2 represents those cells that are cycling as opposed to, cell state 1, which would be non-cycling?
I mean, yeah, that can be a possibility. We haven't explored that, but we will.
I only ask because the transcription factors in state 1 are very interesting. They're almost all entirely mesenchymal homeobox genes and E-box proteins, and those are classically developmentally patterned. And so I think there's a lot more interesting biology in state 1.
Yeah, exactly. Our hypothesis is that the cellular state 1 is in a less differentiated state than the cellular state 2, yes.
I would probably agree with you.
Thank you.
Right over here.
Also a question for Eunice. This was a very interesting presentation. Do you feel that the dual drugged data that you presented, and you said that suggested to you that there may be components of both cell states in individual tumors, but do you believe that there's some plasticity there, and do you have plans to get at that question somehow?
Yeah, I mean, we have that question, but it's a really hard one. Yeah, we think that these cells have a lot of plasticity, but we need to find a way to test how one cell can move from one cellular state to another one. Yeah, we have been trying knocking down transcription factors, or knockout, or also drug treatment. We are still working on that. Yes, great question. Thank you.
Just a quick comment on that. Some data from our lab that was recently published. We didn't show it in the paper, but it showed similar results to what Dr. Fuentes had, in that there's a lot of heterogeneity. And we did see a lot of cycling within the tumors, and those might actually be the gray cells that you're seeing in your single cell. We showed that they kind of start with cycling and then they differentiate to one or the other. But my question actually is for Dr. Mutzayers. I wonder, so you showed some really cool experiments where you were kind of modeling the lung environment versus the bone environment based on stiffness. I'm wondering if you also tried modulating the oxygen tension within those experiments. Because, I mean, our metastatic osteosarcoma tumors are actually, I mean, they're often within the alveolar space. They're at an area of the highest oxygen tension. And if you look at them either at a transcriptional profiling or imaging, they have just massive amounts of mitochondria. And we've often wondered whether that might be one of the reasons that they love that oxygen-rich environment so much. But they might respond differently, not just to the stiffness, but to the oxygen tension.
Yeah, that's a great question, because we set out to look at both variables, but we didn't combine them in those experiments. And so those hydrogel experiments were done exclusively in room air, but we can, and even to speak to the hypoxia experiments, that was in a set 1% versus room air, and you can debate about the realistic nature of, all of the cell culture work that's done in room air, and even whether 1%. And so we're going to move towards more controllable hypoxia and glove box type based experiments where we can program that and be able to do as you've suggested, which is expose those hydrogel stiffness experiments in that kind of hypoxic environment, whatever one we choose. And speaking to yesterday's talk, hydrogels, we heard yesterday from the Stanford work about how you can model stiffness in better ways than with hydrogels. We really got into that through our collaborators who look at mechanical signaling and YAP and things like that, who provided the hydrogels with us as an opportunity for us to look into it with that model.
And I'll just point out that they're actually, we put out a paper recently where we're just simply exploring heterogeneity. And within the heterogeneity of the tumor cells within the lung, one of the things that was most striking was heterogeneity within these metabolic pathways. So this was all transcriptional data, but there were definitively cells that had clear transcriptional programs for glycolysis versus anaerobic metabolism within the same clusters, and even in the tiniest tumors within the lung, where there couldn't possibly be diffusion restriction.
So Ryan, you saw like hypoxia-inducible type of signatures as well? HIF1 signatures?
Yeah. No, we did. Yeah. It was a traceback experiment, and we saw clear evidence of some sets of cells that were undergoing hypoxic type metabolism, even within the lung, under these high oxygen tension environments, and others that were undergoing anaerobic metabolism.
It looks like it for right now.
All right. Well, thank you to our first 4 speakers. We're going to move into the second half of our panel discussion with Dr. Van Heist.
Okay. Hi, everyone. I just first want to thank Christina and Anne and everyone here at MIB Agents for inviting me again this year to speak. I'm very grateful to be here. As Dr. Davis said, I am a pediatric oncologist that does translational research, and so I'll be presenting some basic research that we've done in the lab. And then I also write clinical trials, and so I have some exciting new updates from Factor of last year. I will be discussing the use of an investigational drug called TEW7197, also known as Factosertib, that's manufactured by Medpacto. So, as probably does not need to be said because it's been said countless times here, molecular targeted therapy approaches for the treatment of osteosarcoma are just not feasible due to the complex genetic heterogeneity of the disease. So that's why many of us are looking to target the tumor microenvironment. The osteosarcoma tumor microenvironment is comprised of many host-derived cells that, taken together, favor the development of chemoresistance in the primary tumor, as well as the generation of metastases due to the generation of a permissive niche at both. So literature suggests that TGF-beta is critical in the development of osteosarcoma. It's an important immune suppressive molecule in the tumor microenvironment. Its expression has been shown to be increased in patients with osteosarcoma compared to healthy controls. And its increase or production correlates with high-grade osteosarcoma and is associated with the presence of lung mets. And the literature also has shown that TGF-beta signaling in myeloid cells is required for tumor metastasis. So trials using antibody-based therapy for the inhibition of TGF-beta have been attempted, but unfortunately these drugs are associated with significant side effects, primarily cardiotoxicity. The drugs themselves also have a long half-life, which overall prolongs their global inhibition. So really what would be more ideal or feasible is to use a small molecule that's orally available, that is easily titratable, has a shorter half-life, and then hopefully less toxicity. So the hypothesis of the lab is that targeting TGF-beta in the tumor microenvironment of osteosarcoma can either reduce and/or ameliorate disease. There are 2 aims, aim 1 of which is still ongoing, but that we initially shared at Factor last year, was to examine immune-mediated anti-tumor effects of TGF-beta inhibitor with immune checkpoint blockade using a humanized immune mouse model. And then the second aim was to develop a phase 1/2 clinical trial based on our data using TGF-beta inhibitor. So what is Vactosertib, or TEW7197. It is a highly potent and selective small molecule inhibitor for type 1 TGF-beta receptor. Type 1 TGF-beta receptor is also known as ALK5. Out of the type 1 receptors, ALK5 is known to be the most specific for TGF-beta ligands. ALK5 is thought to, activated ALK5 is thought to cause phosphorylation of SMAD2 and 3. And when looking at pharmacokinetics against phosphorylated SMAD2 and 3 after exposure to Vactosertib, 4 to 8 hours later, there's complete suppression of TGF-beta signaling, followed by a washout period and return of that signal within 24 to 48 hours. So it has a relatively short half-life. In adult studies that have used Vactosertib for other malignancies, it is very well tolerated. And again, if someone does experience a toxic side effect, that's usually resolved with stopping the drug in short order of time. The drug has shown in animal models to reduce cancer cell migration and invasion, as well as metastasis, particularly with respect to lung cancer and melanoma. So our preclinical data is done in Alex Wong's lab. I inadvertently forgot to include a picture of him, but if you were here for the last panel, you got to see his face for quite some time. The other members of the lab that work largely on this project is mainly Sung Hee, as well as Jay and Suzanne. So in the interest of time, I will forego data that I already presented at Factor last year, but this is a summary of that. We wanted to in vitro to expand on our data from before to look at the effects of Vactosertib on osteosarcoma proliferation. And so what we did was we incubated various murine and human osteosarcoma cells with varying concentrations of Vactosertib. And what we showed was that growth of murine and human osteosarcoma cells were significantly inhibited by Vactosertib in a dose-dependent manner. To verify whether these growth inhibitory effects of Vactosertib was mediated by TGF-beta signaling, we looked at phosphorylated SMAD2 in the murine and human osteosarcoma cell lines, and what we found was that at a dose of 100 nanomolar of Vactosertib, there was complete suppression of this, and then at a dose as low as 10 nanomolar, there was inhibition of TGF-beta inducing phosphorylating of SMAD2. So to determine if Vactosertib had any direct effect on tumor growth in immunocompromised animals, we injected NSG mice with human osteosarcoma cells subcutaneously. And then on day 67, we started treating them with either vehicle or Vactosertib. By day 112, 100% of the Vactosertib-treated mice were still alive, as opposed to 20% of the mice treated with vehicle. The mice treated with Vactosertib also had reduced lung mets. Western blot also demonstrated that phosphorylated SMAD2, as well as c-MYC mRNA expression, was inhibited in the Vactosertib-treated tumors. We repeated the experiment using an immune-competent model, which is the BALB/c mice. We injected them again sub-Q with K7M2 cells, and then starting on day 11, we treated them with either vehicle or Vactosertib. On day 39, after injection, we euthanized the mice, and we examined their tumors as well as weighed them, and in picture D, you can see that's a pictorial representation of what the tumors looked like when treated with Vactosertib versus vehicle. So since TGF-beta production correlates with high-grade osteo as well as pulmonary mets, we wanted to investigate the effects of TGF-beta inhibition in pulmonary osteosarcoma. And as you can tell, we injected mice, BALB/c mice again, treated them with Vactosertib as well as vehicle, starting at day 7 after injection. And the BLI picture, I think, speaks for itself. K7M2 mice treated with Vactosertib exhibited dramatic inhibition in pulmonary burden, and this was further confirmed by a series of other testing as well as H&E staining of the lung samples. Lastly, several studies have examined PD-L1 expression in osteosarcoma tumor cells as well as tissue and has found an increase in PD-L1 expression in lung mets. And so we wanted to look at the combination of Vactocertib in addition to immune checkpoint blockade using anti-PD-1 and anti-PD-L1 antibody. And what we found, we injected BALB/c mice again with K7M2 cells, and we started treating them on day 21 with Vactocertib, and then weekly anti-PD-1 or anti-PD-L1. And what we found after doing BLI weekly for 11 weeks was that Vactocertib, based on our preclinical data as we expected, reduced osteosarcoma tumor growth, but there was not actually much and there was no significant synergistic effect between the Vactocertib and immune checkpoint blockade compared to Vactocertib alone. There was also no synergistic effect between Vactocertib and immune checkpoint blockade when it came to the induction of NK cells or the suppression of M2 macrophages compared with Vactocertib alone. Okay, moving on. So Vactocertib is manufactured by MedPacto. And we collaborated with them to develop a phase 1/2 clinical trial. I am running out of time. So main update since Factor 2022 is that we completed the trial development in October. In January of this year, Vactocertib was designated as an FDA fast track item. We received protocol review and monitoring committee approval at our institution for the trial. We've written a paper that's currently being reviewed. And we are just awaiting IRB submission, which was delayed due to logistical issues. The trial will be a multi-center, multi-continental trial. There's currently 19 sites across North America, Asia, and Europe, and the Center for Pediatric Immunotherapy at Rainbow Babies will be the lead site, of which I am the PI. And to date, several patients nationally and internationally have received compassionate use of Vactocertib. If you need to reach me or our clinical trials unit, this is our information. You can also find it on clinicaltrials.gov. I frequently answer calls, emails from patients, families, and physicians, and I'm happy to do so. Current directions, what we're currently working on in the lab is, as I said, AIM-1, using discarded thymic tissue following cardiac surgeries to create humanized immune mouse models to further examine Vactocertib alone as well as in conjunction with immune checkpoint blockade, and then using TGF-beta inhibitor in addition to cryoablation. I just want to thank again Alex for being a wonderful mentor, all of my funding, my family who doesn't know what I'm talking about but still cheers me on anyway. And I am here because of Charlotte, and I want to thank the Murdoch family for their generosity in funding these efforts. So thank you.
All right, well, that's going to be some great talk to follow up, Kristen, and it's nice that she presented right before me. Thank you to the MIB for the opportunity to talk, and I'm also more thankful that I'm not Nadia, who's the last one of this whole session. So, but yes, let's get started. So I'm going to be following up a little bit with regards to what Kristen was saying about some of the immune checkpoint stuff. And one of our goals is, as everyone knows here in terms of ICIs and sarcoma, has been pretty poor response, obviously, whether it be single agent or even combination immune checkpoint approaches. So one of our projects in the lab is how do we potentially improve this approach, and we're looking at targeting oncogenic signaling pathways that can alter both tumor intrinsic as well as extrinsic microenvironmental factors. You heard a little bit about what Betsy was mentioning as well with the sting. So this is just some old data you guys have probably seen with regards to ICIs and sarcomas. So oncogenic signaling pathways, obviously you have a lot of receptor tyrosine kinases that activate internal things such as proliferation, invasion, metastatic type signatures. And then also another pathway of our interest is the beta-catenin Wnt signaling, beta-catenin signaling, easy for me to say, which is just diagrammed on the right-hand side. You can see basically a transmembrane receptor activation by the ligand. You get beta-catenin gets dissociated, it translocates to the nucleus, binds its transcriptional complex factors, and turns on downstream target genes such as c-Myc, cyclin-regulating proteins, and other things that are often involved in metastasis and stem cell-like properties. But of course, these pathways are just not involved intrinsically, but they also have potential extrinsic effects, such as on the tumor microenvironment. So on the left-hand side, the AXL MET signaling pathway is one of the things we're focusing on, but we're also looking at other pathways as well. You know, the AXL MET, with regards to extrinsic effects, could have, you know, alters VEGF signaling in terms of expansion to MDSCs and regulatory T cells, could suppress MHC1 expression, as well as increase some of the exhaustion markers. And on the right-hand side, Wnt signaling has also been shown to be involved in tumor immune cell exclusion, so anti-tumor immune evasion. And so on the lower left-hand part of that right-hand panel, you have Wnt signaling that's involved in inhibiting tumor antigen release. Then as you go towards antigen presentation, as you cycle clockwise, tumor T cell priming and activation, T cell infiltration, and so forth. So obviously these pathways are not only important from an extrinsic standpoint, but also potentially modulating the tumor microenvironment. So two of the drugs that we are interested in are cabozantinib, as well as tegavivint. So on the left-hand side, cabozantinib is just highlighting some of the pathways it's targeting. Of course, like the VEGF pathway, the MET, AXL, things like that. I already went over some of the tumor intrinsic aspects that are involved with regards to targeting that cabozantinib targets. On the right-hand side, you may not be as familiar with, is tegavivint. I was really excited to hear Giovanna yesterday mention tegavivint as part of one of her molecules of interest, or came up on her list as potentially targeting as within the master regulators of osteosarcoma. So we've been working with tegavivint for several years now. It's a drug produced by Therapeutics. So basically, it's blocks the interaction of TBL1 and beta-catenin. So on the right-hand side in that schema, you have TBL1 beta-catenin, which is required, kind of helps beta-catenin translocate to the nucleus and then obviously turn on its downstream target genes. Tegavivint blocks the binding pocket with regards to the TBL1 beta-catenin interaction and leads to beta-catenin degradation. We've gone through the mechanism. We've published this in a previous paper, and I'll show you on the next one. So why are we interested in these, of course, drugs? Of course, we know cabozantinib has had the Cabo trials showed obviously some encouraging results for osteosarcoma patients, and of course now cabozantinib is being incorporated into an active pediatric clinical trial for newly diagnosed osteos. And tegavivint on the right-hand side. So as I mentioned, we've been working with this drug for about 5 or 6 years, interacting with the company. We did a lot of our lab, as well as other labs, did a lot of the preclinical data. We published this back in 2019 in the JNCI, showed it was very effective in targeting osteosarcoma and metastatic osteosarcoma. We then worked with Iterion and COG to come up with the clinical trial. Dr. Sarah Whittle, who's at Texas Children's, where I was at TCH up until last year, is the chair of this particular trial, which opened about almost 2 years ago. So obviously it has pediatric relevance. So you heard from Anthony about genetic engineered mouse models. My lab has produced a lot of GEMs as well for sarcomas, including osteosarcoma. We have rhabdomyosarcoma. We have other genetic engineered mouse models. So we have a conditional model as well with a p53 alteration. But we've also more recently generated a hyperactivated c-MYC model. We wanted to do that because, again, c-MYC amplification and hyperactivation of c-MYC is thought to be associated with a poor prognosis, obviously, in pediatric patients. It's a potent oncogene. And so we've derived several syngeneic murine cell lines from these particular models. I was also excited to see from Anthony's data, obviously the metabolic processes seem to correlate pretty well between the murine, dog, and mouse. So that was great to see. So just getting to the study design real quick. So we did the Cabo. First off, I'll tell you about the Cabo results, and then I'll show you some of the tegavivint results. So again, these are preclinical studies using Cabo. This is the study design. We can take our syngeneic model. This is a p53-based model only. And we can inject it into the tibia of the mice, of the C57 black 6 mice. We do not label these, unlike, unfortunately, with Kristen's approach, which was really great with the luciferase. We cannot label these cells with luciferase or GFP because actually those are immunogenic in our approaches. So the C57 black 6 mice will actually reject those tumors. And so these are blinded, non-labeled cells. So we inject them to the tibia, let the tumors become palpable, and then we obviously do a pretty comprehensive, we can form our own little, obviously, clinical trials, as kind of Kristen showed with her TGF-beta inhibitor, but we can obviously do our own clinical trials with obviously the appropriate controls, the single agent therapies, and then the combination therapies. And we actually tested 2 ICIs, one being TIGIT and one being anti-PD-L1. And then at the end of the study, we assessed, obviously, primary tumor and looking for potential metastatic effects. So first off, these are just some of the quick results. Obviously, I don't have time. Oh, good, I still have 10 minutes. This is great. I love this clock. I was speaking really fast because I figured I haven't looked over there, but I still have 10 minutes. On the left-hand panel, again, what I want to highlight here is a couple of things. If you look at the tumor volumes on the left-hand panel, so this is kind of a nice little summary of all the models, of all the cohorts that we did. So we obviously did the appropriate controls, the single agents, and the combination therapies, even combination of the 2 ICIs. So there are a couple, I think, take-home points here. In our mouse models, and I can show you this for other cell lines we use, this is highlighting the F420 cell line, but it's true about other syngeneic models we've tested. Single agent, and even combination ICIs don't really have an effect, all right? So I think that kind of correlates to what obviously we see in the human patients. So it's not like everything works in the mouse. This is showing you that these drugs, these single agent at least, and combination ICIs don't really have an effect on our osteosarcoma tumor cells themselves. Obviously, in this particular model, the Cabo had a very good response, as the third column there. And then when you highlight, if you look at the middle panel, we're actually comparing the Cabo alone to the Cabo with the ICI. And we do see a statistically significant difference in tumor volumes at the end of this particular study, especially more so with the anti-TIGIT. Interestingly, we did actually look at, do some kind of a targeted gene expression analysis on Cabo-only treated tumors, and that's shown there on the right-hand side. And what you see here is some upregulation of what's called Th1 type of response genes that are associated with T-cell infiltration. And then we also see a decrease in immune evasion type of signatures, TGF-beta, your CCR2, which you heard from yesterday from yesterday's talks. So again, we're seeing signatures that are associated with potentially making it a more immune-friendly microenvironment. Let's see, where am I going here? Oh, wrong one, sorry. So, and then we also looked at these particular models and looked at gross metastasis. We still need to do some micrometastasis, but what you can see here is that the Cabo only, if you look on the right-hand side, the Cabo only treated mice still had evidence of metastasis in these orthotopic models, but with the combination therapy with the TIGIT, we didn't see any evidence of gross metastasis. We still need to do more comprehensive studies on this. Oh, now I only have 2 minutes. That's not fair. And obviously with this model, we showed that it was very sensitive to Cabo. Interestingly, one of the reasons why we generated the CMYC model, not just because we're interested in understanding the biology a little bit better, but also these provide interesting additional models. On the left-hand side, you can see the kind of the cytotoxicity assay curves for Cabo in the non-MYC driven, which are the red, versus the CMYC hyperactivated models. You can see the CMYC hyperactivated models are much less sensitive to Cabo, and that also actually translates from in vitro to in vivo. You can see on the right-hand side in blue are the MYC hyperactivated models, which again, pretty much, I don't say blow through, but they grow through pretty rapidly the cabozantinib treatment that was pretty effective in the F420 model that I've just previously showed you. So now we're actually doing further studies on these more aggressive, potentially more resistant models to get mechanisms of resistance, as well as potentially model, hopefully, what some of our hyperactivated or MYC amplified patients that we see that often relapse. All right, with 1 minute left, I got to zip now. So the tegavivint is the beta-catenin targeting agent, and I just wanted to show you real quick. So we did, again, using our syngeneic models, we saw that tegavivint had an anti-tumor activity, which is shown on the left-hand panel. But what we did is that we actually isolated the tumors at the end of the study and did bulk RNA sequencing on these. And what I want to highlight is in this pathway analysis is that if you look at the blue, the bottom kind of pathways, or the blue highlighted pathways, on the right-hand side, what do you see that's downregulated by the tegavivint? So we see Wnt signaling, canonical Wnt signaling. So we have in vivo evidence that the tegavivint is targeting the pathway of interest. In addition, complementing with Kristen's work, obviously, as well, we also saw a significant decrease in TGFR beta signaling, or receptor activation. So those pathways are really decreased in our tegavivint treated. So in the red tumors versus the black tumors, our tegavivint treatment tumors are decreased in these oncogenic and more tumor-promoting pathways. But what's activated? What's hyperactivated? And that's shown in the red color. And you can see here a lot of the immune cell signaling pathways are upregulated. Natural killer cells, T-cell receptor signaling, and so forth. And finally, we also then showed that we see a higher infiltration of T-cells, CD8 T-cells, in the tegavivint treatment tumors, and that's by IHC, and by flow we kind of confirm that. We see higher CD8s as well as a higher NK cells infiltration with the tegavivint. Last two slides. So tegavivint induces an anti-tumor immune signaling response. So this is one of the other advantages of our models, we can actually do these real-time correlative biology studies. So we can take our mice, we can be treating them, and we can obviously isolate blood or blood plasma from them during their treatment and at the end of the treatment course. I'm showing you this is data at the time of sacrifice, but we can also do longitudinal studies during the treatments. And so we just did this and sent it off our cytokine panel from the blood. So we compared control versus tegavivint treated mice in 2 different models here, but we've also seen this in other models that we've studied from our syngeneic gems. And you can see here IL-4 goes down significantly in the tegavivint treatment, so IL-4 inhibits anti-tumor T-cell activity. And one of the interesting cytokines or chemokines that went up is CXCL9, which promotes anti-tumor immune response. And what's interesting is that in human patients, in human melanoma patients that have high circulating CXCL9, they've been known to respond to checkpoint inhibitors. So we just did the study, of course. We did the appropriate studies where we took our syngeneic models. This is, again, showing results from 2 of them, where we did the appropriate controls, the single agents, and the combination with the ICI. On the left-hand side is a less aggressive F331 model, but you can see here that the combination does slow down and basically hold the tumor volume in check. I wish, of course, it regresses, as Ryan Roberts mentioned yesterday. We're always hoping for maybe only following up with things that are really regressing the tumors, but we're optimistic here and following up on future studies here. On the right-hand side, a more aggressive model, the F420, again, shows that the combination has some potent anti-tumor activity. The last thing I want to show here, though, is that in that combination group here, that cohort, that purple group, we actually had a few mice left over, and what we saw there when we let go of the treatment after 4 weeks is that the tumors basically tripled in size within 1 week period of time. So, of course, that's stopping all therapy, but we had about 3 mice that were remaining in that one cohort. And, obviously, so indicating that the combination therapy was doing something to hold it in check. We are now following up with repeating these studies, but also following up with these from a molecular standpoint to identify potential mechanisms of resistance and obviously sensitivity using these type of model systems. I won't go over this, but we're also combining these with other immune modulatory agents as well as adoptive cell therapies we're working with in terms of using these targeted therapies. And of course, I'd like to thank all of the lab members and my support, obviously including MIBG, and thank you very much.
Good afternoon, everyone. My name is Nadia. I'm a research associate at Sarcoma Oncology Center, Santa Monica, and it's a great honor to be here at this wonderful meeting. And I'm very grateful for Megan's family who funded my travel award. In lieu of Dr. Gordon, today I'm happy to present our work. Let me start. Let me start with the definition. What is delta-RxG? Well, delta-RxG is a Maloney-Murine leukemia virus-based tumor-targeted retroviral vector displaying high affinity to signature SIG or collagen-binding peptide. Off the shelf, injected intravenously, delta-RxG seeks out and accumulate in tumors by binding abnormally exposed collagenous proteins in tumor microenvironment. And in tumor microenvironment, the vector binds, infuses, and integrates into the chromosomes of rapidly dividing cells, such as cancer cells, and not in the normal cells, since normal cells don't express the abnormal protein. In the mechanism of action of delta-Rx, in cell culture, Delta-Rx induced apoptosis of cancer cells, and in animal studies, Delta-Rxg inhibited tumor growth in a nude mouse model of osteosarcoma. Mechanistically, delta-Rxg blocks cell cycle in G1 phase, and as you can see in a D picture, cysteine G1 is highly expressed in tumors, and in e-picture, you could see how CCNG1 expression downregulates by delta RXG treatment. And what is cyclin G1 or CCNG1? CCNG1 is an oncogene that activates by proximal signaling pathways, you can see in the slide. And by activating, CCNG1 activates MYC-MDM2, which leads to inactivation of TP53. So by inactivating CCNG1, we can restore TP53 tumor suppression function. Delta RXG in vivo targeted gene delivery system that can be taken out from the freezer, turned in the water bath, and infused over 30 minutes. And delta RXG doesn't cause the side effects, which is usually caused by standard chemotherapy, like hair loss, nausea, vomiting, diarrhea, myelosuppression. The only adverse events we observe are like hypersensitivity, infusion reaction, which could be prevented by dexamethasone dihydroxypyramide. So delta RXG has been tested in 4 phase 1-2 studies in osteosarcomas, pancreas cancer, osteosarcomas, and breast cancer, and engaged orphan drug status in osteosarcoma for soft tissue sarcoma for pancreas cancer, and fast track designation for pancreas cancer from FDA, US FDA, and also accelerated approval for all chemotherapy-resistant solid malignancy by Philippine FDA. So in 2013 was reported that the CCNG1 highly expressed in cancer stem cells, and delta RXG as the CCNG1 inhibitor could eradicate the cancer stem cells, which could prevent recurrence and metastasis. And in a small phase 2 study of delta RXG, long-term survival study showed 3 of 18, it's almost 17% patient with chemotherapy-resistant advanced osteosarcoma. They were alive in sustained remission more than 14 years from delta RXG treatment initiation. And so our hypothesis, so CCNG1 is a novel biomarker. They may identify patients who will respond favorably to delta RXG therapy. We use Boston gene artificial intelligence approach that integrates genomic and transcriptomic analysis of tumors and tumor microenvironments to reveal key drivers in tumors such as immune microenvironment properties, actionable mutation, biomarkers of response to diverse therapies, and generates a personalized roadmap for therapeutic decision making for individual cancer patients. This is the method for CCNG1 RNA analysis. So an integrated analysis shown tumor molecular portrait, which divided into 4 types: immune-enriched non-fibrotic, immune-enriched fibrotic, and immune-desired. And as you can see, first 2 immune-enriched tumor microenvironments have been shown to have a better response to immunotherapy in comparison with second 2. So the Boston gene is the only company that reports CCNG1 expression levels in sarcoma. So we divide them by high, medium, and low level. Here you can see the enhanced CCNG1 expression in all tumors of patients being followed at our cancer center, and specifically for osteosarcoma. You can see that the osteosarcoma mainly has the medium level of CCNG1 expression by our experience. This slide shows the reduction of 25% reduction in size of the palpable chest tumor of patient who was on Delta-Rex, and the reduction occurred after only 4 weeks of treatment, and CCNG1 expression level, particularly for this patient, was 74%. which is high medium. I'm sorry. Yeah, so this slide shows the significant reduction in, so this is the molecular portrait, and it shows significant reduction in a size of tumor. Oh, I'm sorry. Here and here in comparison, from 70 to 30%. And CT scans showed only stable disease, so we assumed that the molecular portrait of tumor microenvironment could be a sensitive indicator for response along with the CT scans. And I wanted to show you the long-term osteosarcoma survivals from our experience. And one of the patients is actually in our meeting here. And case one is the patient was at Delta Rex for 4 months and followed by metastasiectomy, then treated post-op with other 4 months of Delta Rex G in a life of 15 years. Second case, patient was at Delta Rex G and Delta Wax for 6 months, followed by four cycles for doxorubicin in a life of 14 years. And case 3, Delta Rex G monotherapy for 1 year, followed by surgical resection in a patient in life for 14 years. And in a conclusion, we wanted to say that the precision medicine strives to find a target oncogenes from many oncogenic drivers from different histological subtypes. But in addition, Dr. Gordon believes that we should strive to identify target oncogenes that are precisely common to different histological subtypes. And we suggest that SCCNG1 is the common to all types of sarcoma. In a multi-center phase 2-3 study for sarcoma and osteosarcoma in place at our center, and using Delta G-Rex as the platform therapy in a combination with immunotherapy and targeted therapy following metastasectomy. Primary endpoint going to be progression-free survival, correlative analysis of response with CCNG1 expression level in tumors of Delta Rex G. Treated patient will be done, and qualitative and quantitative analysis of ctDNA will be monitored as a response to therapy. We don't have any conflicts of interest. And well, I'm very thankful for Dr. Gordon for everything she taught me, for her kindness and support. And we also grateful for our benefactor of AVNE foundations, and especially Megan Holmes Memorial and Lichowski Memorial. Thank you for your attention.
Thank you to our panelists for a really excellent final panel of this Factor conference. And we'd like to open the floor to any questions. Anybody?
Any questions?
I have a question for Dr. Van Heist. I'm right here. I was wondering what, congratulations on almost getting the trial open. What do you think is the next step? Looking 2-3 years down the line, what are we going to add to Vactosertib?
That's a good, oh, we're not on, whatever, I can yell. Hello, okay. That's a great question, and we do want to expand beyond just monotherapy of vactocertib if that's possible. And so right now in the lab, I'm working on using vactocertib in addition to cryoablation. I mean, we literally just started like 2 weeks ago, so I can't say much about that yet. But we've also, like I mentioned, we are trying to create a humanized immune mouse model just to trial vactocertib in addition with immune checkpoint blockade in that model. And I know we frequently get asked pairing vactocertib with chemotherapy, and we haven't trialed that yet, but I think it's a possibility. Certain chemotherapeutic agents like Cytoxan in particular have been shown to activate myeloid cells, and so it's not so much that we think the two would be good combination for like causing tumor damage, but rather just may work synergistically to activate the immune system. So, but again, it's like a battle of like trying to minimize side effects as well for patients. So we definitely have a few ideas on the horizon. We've also talked about NK cell therapy in addition to vactocertib. And so hopefully, maybe at future presentations, I'll have more to share.
To your left, mid.
Brian Ladle from Johns Hopkins. Thanks for the great talk. Maybe 2 questions. Kristen, with the vactocertib and I don't know, can you expound a little, like what do you think as far as direct anti-tumor effect versus, for us it's interesting that vactocertib actually inhibits tumor cells directly. So the effects you think, do you think it's mostly direct tumor, or is it more tumor microenvironment and the immune suppressive effects? Then I had a question for Jason as well.
Okay, in my opinion, and Alex is here as well, so since I'm not innately a basic science researcher, but in my experience in working on the lab in this project, I think it's mostly with respect to the tumor microenvironment, not directly on the tumor itself. We've shown that like in animal models that have more significantly bulky disease, the vactocertib, while it will slow down progression of the tumor, it doesn't work as effectively as if the tumor is already smaller to begin with. And I think just like in a clinical scenario, it would probably be more helpful to patients who don't have as bulky disease burden. And I think, again, that also plays into account it's like what's going on in the tumor microenvironment. So my opinion is that that's where it's more efficacious, not on the tumor itself.
Thanks. And Jason, just so you did a combination with TIGIT. Any specific reason to choose that one in your model, and also the beta-catenin inhibitor, right? Do you think there might be a different inhibitor that would play along with that one better?
Yeah, so that's a great question, Brian. Thanks. So one of the things we looked at the TIGIT because we knew it had we checked our models for expression, so we knew that there was TIGIT expression. Plus B, there also was a Nature Communications paper just a couple of years ago showing that TIGIT is present in a good number of osteosarcoma models. So we wanted to obviously overlap that as well. We're trying to model that. Now, in terms of the combinations, we're exploring other combinations, as well. We're also exploring other combinations with just tegavivint independent of the ICIs because we think that we're pretty excited about that particular pathway in terms of its potential for targeting, and it's so far been very safe in the phase 1 trial. And so we've done a high throughput drug screen looking for synergistic combinations.
We've heard about some very interesting molecular targets and all very kind of separate discussions, of course. Have you, through being on this panel and hearing about these other potential targets, have any of you come up with ideas that you want to test back when you return from Factor? Some enzalutamide plus some cabozantinib, I don't know.
No, I think it's been obviously a very stimulating conference, I mean, hearing even the last panel in terms of also the, the anti-CD47 in combination with the ICIOs, for the dog, approach is really interesting. So there may be other cocktails that can come up. And I think that's one of the advantages, at least from our mouse modeling standpoint. We can set up so many different approaches and combinations and at least see if there's any inkling of, potential to further investigate. So, I mean, we can, within 2 months, we can have an answer to, at least some insights into whether a single agent or obviously some combination therapy looks potentially further, promising to further pursue.
One more question back here to the right, in the back of the room.
Thank you again for all the presentations. Back to the TGF-beta question. Have you looked at, TGF-beta inhibitors have been tried in a lot of adult cancers, and majority of them have failed, unfortunately. And I'm a big believer in TGF-beta inhibitor, but I don't think it's going to work by itself, or maybe not even the immune checkpoint. Maybe other things need to be done. But have you tested this in galinosertib, for example, some of the other small molecules for TGF-beta inhibition to see if it gives you the same result? Is this a drug that's slightly different than traditional TGF-beta inhibitors? And if the trial fails to reach its primary end goal, do you think this is the end of this drug, or is there plans to do other combinations to try it out? Because currently there's no TGF-beta inhibitors FDA approved, as far as I know.
Yeah, so I'm sorry, the lights are sort of impairing my ability to see you, so I'll talk in that general direction. Yeah, at the beginning of my talk, I mentioned that there had been trials done with different therapies to target TGF-beta, and they largely fail, in my opinion, due to the side effects as well as the fact that the drugs have a longer half-life. And so they just overall have prolonged global inhibition. But with respect to the other TGF-beta inhibitor that you mentioned, Vactosertib is actually 10 times more potent than that drug, and it also has been shown to have significantly less side effects, particularly cardiotoxic side effects. And based on the data generated from our lab, we did get FDA approval, IND approval for the use of Vactosertib, and we also just got, based on the data from the lab, Medpacto also just got Vactosertib fast track FDA approval for treatment in osteosarcoma. So I think this drug in particular, compared to others that have been used, is probably more promising in a clinical setting. I hope that answers your question.
No, that's great. I guess follow up to that is do you think the PK in the mouse studies are similar to the humans? And when you say short, which is great, but if you take the drug off for a couple of days, do you see phosphorus SMAD activation quickly following?
So with respect to humans, the Vactosertib itself has actually been used in human trials, in adult trials, for a variety of malignancies, and not osteosarcoma. And the drug has been well tolerated in human population. There are several pediatric adolescent aged patients are in current compassionate use of the drug nationally and internationally, and it seems to be well tolerated, but we obviously haven't studied what you're mentioning with regard to phosphorylation of SMED in them, because we don't have the trial open yet.
Thank you.
I believe that was the last question of the conference.
All right.